Good afternoon, everyone, and welcome to this webinar on advancing in silico respiratory medicine. This is an introduction to a series of joint activities our EPSRC-funded network BioReam have planned alongside In Silico UK and is supported by NIHR and the Manchester Respiratory Translation Research Collaboration. So just to give you a quick rundown on our very action-packed agenda this afternoon. First, I will give you a brief introduction to BioReam, the network. I will then invite Professor Alex Frangi, who is chair of In Silico UK, to talk to you about what they're doing within In Silico UK, followed by a presentation by Professor Salman Siddiqui, who will give a clinical perspective on the areas where In Silico technologies will play a key role in respiratory medicine. Alex will then come back to describe how the joint activities we've planned will play out. And finally, we'll open the floor to a Q&A session, which will be led and moderated by Thierry Marchal, who is president of the Avicenna Alliance and CTO of Healthcare for Europe at ANSYS. Please do feel free to put questions in the chat as we go along. We'll address them in the Q&A session at the end. Okay, so what is BioReam? It's an EPSRC funded network plus. Currently, we're an international community of over 250 researchers from multiple disciplines representing multiple institutions, industry, and charities. Our common interest is in advancing respiratory medicine. We've been going about 3 years now, and it's hosted at the University of Nottingham, chaired by myself. My background is in developing multi-scale models of asthma in close collaboration with clinicians, experimental biologists, bioengineers, and computer scientists. And I think I failed to mention my own name. I'm Bindi Brook. I'm a professor of mathematical medicine and biology. And the network co-chair is Professor Alex Horsley, who is a clinician and researcher who specializes in cystic fibrosis. The other clinical theme lead who we'll hear from later on is Professor Salman Siddiqui from Imperial. And the leadership team also includes Professor Peter Robbins from Oxford, Professor Declan Bates from Warwick, Professor Nick Hill from Glasgow, and Professor Jim Wild from Sheffield. Okay, so we essentially started this network. One last thing I should point out as well is that we have been working closely with Asthma and Lung UK, the NIHR and biomedical research councils across the universities, and Innovate, In Silico UK, and more recently the Avicenna Alliance. So a brief background to the network. We started this because many of us felt that although we were doing research to advance respiratory medicine in our different ways, it was all happening in our own silos. So sophisticated modeling, advances in imaging, cutting edge experimental medicine, and of course, AI. But we felt that this siloing was hampering progress and that if we really wanted to advance respiratory medicine in a transformative way, then these cutting edge tools, techniques, and technologies really need to be developed in truly interdisciplinary frameworks and translated into the clinic and industry. So our vision is to break down barriers to clinical translation of in silico respiratory medicine through our commitment to interdisciplinary collaboration and cutting-edge research. The network is built on these 4 research themes or priority areas. So improving patient outcomes by improving treatment delivery and clinical trial design, detecting disease early by developing the next generation of clinical lung function measurement, improving chronic and critical care approaches through modeling pulmonary mechanics, and truly understanding the impact of environment on lung health across the scales. In order to realize our vision to translate research into clinic, a key forward-looking step for us is to ensure that we build the right kind of evidence base that will be accepted by regulatory bodies at the outset. That is while we're doing this collaborative research. So that's why we've organized this series of events and essentially where In Silico UK comes in. I won't go through these points separately because we'll essentially be discussing these different aspects of this this collaboration and this series of events. And in particular, this is where In Silico UK comes in. So at this point, with that very brief introduction, I will hand over to Alex Frangi, who will describe in greater detail how we might achieve some of these aims that I list here. So if that's okay with you, Alex.
Yeah, thanks a lot, Bindi, and good day to everyone. My name is Alex Frangi, and I'm professor at the University of Manchester. Everything is okay. You should be able to see my slides now.
Yes. Thanks.
So as well, before I start my talk, I should say that we are very thankful to our funders and collaborators, and in particular to UKRI, the Medical Research Council, and Innovate UK, and MHRA, the Medicines Health Regulatory Agency. That is, we are working very closely in this area of regulatory evidence. And the topic that I was given was current progress and barriers. I sort of slightly modified it into barriers, enablers, and current progress to show that there is actually challenges, but those are not the last word. There's a lot going on. And I'm really delighted that we developed this joint webinar with BioRim, because our mission, we as a network are organ system agnostic, but we are looking at across the whole medical products spectrum, but we are very interested in working vertically with different networks in different disease domains. And this is a great example of this collaboration and cross-council collaboration as well. So first, I'd like to explain you why we think we need in silico technologies and why we need this digital evidence for this digital era. This is something that if you come from the network BioRim, I know I'm going to be preaching to the converted, but I think it's useful to understand some of the underpinnings of why we do what we do, right? The first rule in medicine is first do not harm. And this is the Hippocratic oath that every clinician, Salman himself, would have taken at some point in his career. But let's reflect on this and how we are doing at the moment. So the whole purpose of medical products is to support clinicians in delivering the best health possible to patients. And the way we develop products in medical products, so drugs or medical devices, is by sifting them through along the life cycle of their development and subsequently regulation, sifting them through a process of different sources of regulatory and scientific evidence. They're aiming at answering two questions: is this product safe, and is this product performant? And we do that through three main avenues of evidence: bench testing, animal testing, and human testing. And I always remind my own students that all three of them, including human testing, they're just models. They're experimental models that are trying to let us to understand how our intervention, by testing it in a small group of people, may broaden its impact to the general population. Now, is this actually working? Because this is the whole edifice of medical products is predicated on these being solid sources of evidence. So for starters, we know that all of these different areas have limitations that I'm not going to go into details, but you'll have the recording of the webinar for it. But there are limitations in all of them. And also there are limitations in general in the way we do science in terms of reproducibility and transparency. We also are unfortunately not safe, which is one of the things that we are claiming that we're aiming at. So a number of studies, the implant files in the US has shown the potential harm to patients, and also the Cumberland report, more specifically in the UK, has highlighted the impacts that our health ecosystem has in delivering the best care, and how in some instances it has actually failed to do so. If you also look to the news, or if you look to the FDA recalls page, you will see regularly situations like this where a particular product is taken out of the market, and it has already produced a number of impacts and harms to patients. But also, in this particular device, as far as I understand, this is the last left ventricular assist device that was in the market. So sometimes we can we need to be safe, but also it means that we are losing life-saving therapies potentially as well. And this is not just this particular company, these particular products. There is a number of recalls across the first semester of 2024 alone is of nearly 49, and pretty much all the key manufacturers have been in one way or another entangled into these processes. Also, thanks to my colleagues Tina Morrison from FDA and Mark Palmer, I'm bringing these two slides, which actually give you an idea about the challenge of the magnitude of the challenge we are talking about, and that there is an ethical imperative regarding patients. So from every 100 complex medical devices that go to for clinical testing, only 30 of them will ever get FDA approval, which effectively means that there is about 13,000 patients or 31,000 patients, about 41% of them, that have been exposed to devices that will remain being investigational and will never see the market. Okay, so these are people that we have in a way potentially harmed. Now, if you look at this from a pharma point of view, the numbers are even worse. For every 100 drugs, there will be only 5 of them that will get FDA approval, and 32,000 patients, or 47,000, that when participating in those trials will have been exposed to drugs that will remain investigational. So it's a big, big problem. Then, in addition, these sort of approaches take forever, 12 months for a typical drug, 7 years for a typical device. They're very expensive, $54 million for a single complex medical device if you get it right first time, but normally we don't. So if you include the capitalization cost, this is about $500 million what it takes on average. And if you see the probabilities of success of moving from one phase to the other phase through the R&D life cycle, you see that there is a 90% attrition rate for devices, and you have a similar numbers for drugs. Another aspect is patient recruitments. Is very expensive. We have lots of failures or uncompleted trials that have essentially not achieved the targets that they started for. And also, quite importantly, sometimes we don't have a good representation of the diversity of patients across the studies on the trial. So we will develop studies that will generate an evidence with the population that then will not be representative of the population at large that we will be exposing to these products. And some, the bigger the number of patients you need to recruit, the higher the cost, and at some point this becomes prohibitive. And that's why if you don't create an ecosystem that is supportive of industry in this sector, in this area, companies will go to other places where regulations or testing is easier or more effective. And that's why our value proposition is how we reduce this R&D time from years to months, and we develop technologies that can fail fast, fail earlier, so that we create the right ecosystem, the right milieu for industry to want to come to the UK. Given that at least both of our networks are funded by the UK, you would allow us to raise the flag of the UK here. So the way that I understand first do not harm is as simulation first. So we need to go and address these problems similarly to what other sectors in industry have done, like the aerospace, where there are very complex systems, where the more complex the systems are, the less reliant they are on physical tests and the more on virtual testing. And while I don't argue that medicine is the same that aerospace, but aerospace is still quite a complex system with mission critical, highly regulated, highly full of uncertainties, multi-scale in nature. So a lot of the similar challenges that I know BioRim is dealing with from a computational point of view. So what we say is we want, as modelers, we cannot pretend that all the evidence will be gathered purely on modeling grounds, but what we can aim is at saying, look, if there is an ethical imperative that if you have 4 sources of evidence, bench, animal, human, and in silico testing, you don't rely purely on 3 of them as we do today. And also that we develop ways to integrate all of these 4 sources of evidence into a way that provides a balanced set of scientific and regulatory evidence, so that we can identify products earlier on through modeling and simulation that will never be able to go all the way to patients with the risk patients and with the risk investments in that way. And we use the usual pathways of human and animal testing more as a validation and confirmatory evidence than as discovery evidence, which unfortunately sometimes we are using at the moment. So industry has seen already that in some sectors, for instance, these are numbers from one particular product, from one particular manufacturer, but you can go 2 years earlier to market, reduce the number of patients you need to expose in a trial, reduce the cost concomitantly related to that, and increase substantially the number of subjects, patients that are benefit from these life-saving therapies by essentially going earlier to market, and of course, indirectly the revenues to the companies. So what in BioRim, you're doing, and what we are doing in other sectors as well, is develop models of virtual patients, models of the interventions, drugs, devices, and models about how do we read out success, basically, or responses and outcomes to those therapies, which are the 3 key ingredients from what in silico trials are. So I would like to propose to you today that perhaps what we need to change is this primo non nocere sort of Hippocratic oath to the digital area, by saying that the second task we need to do is we'll always keep the non-harming to patients as the first objective, but the second thing we should be doing is simulate, and rather than before going to any other forms of evidence. And that's what led us to develop the concept of the Center of Excellence on in silico regulatory science and innovation that was announced a week ago, its funding. But that this Center of Excellence, or CERSI as we call, is essentially the operationalization of the work that we have done in the in silico UK network, through which we've been over the last few months and years. This has been a 4 years journey. We have gone through developing roadmaps, developing evidence from the economic impact of these technologies for the UK, how they have been influencing or affecting private investments through startups, also working with regulators to generate an understanding about what the regulators need from these technologies, and more recently also reaching out our engagement to members of the public through a podcast that we have started, and also through a document that is more oriented towards C-suite people in companies, so that this message is understood also in the best way possible. But there is a lot of work going on elsewhere internationally, so we are by no means the only ones working in this space. And what BioRim and in Silico UK, the CERSI is trying to do is help to promote these technologies within our national ecosystem, but also well connected internationally, to make sure that what we do, contributes to the international harmonization that is required in this space. Now, the last part is about barriers and enablers. And we did some time ago, about 2 or 3 years ago, a questionnaire to about 300 people we got, where we asked them, what are the top 5 perceived barriers for the adoption of in silico trials and the use of computational modeling? And you see that there are 3 things that came topping this list. One is the fact that it's unclear for industry and for academics whether regulators will accept this evidence or not. I think from 3 years to now, we have done a massive progress in that, through to the rich discussion. And in fact, the fact that there is a funding center of excellence on in silico technologies by the government and with MHRA involvement tells a lot. And it's not just MHRA, it's NICE, it's HRA. The other thing is, the second one is the maturity and model credibility. And if I went through the detailed nuance of the comments that we got, what you would see is that what it means is that there are areas where we have very sophisticated and mature models, but areas where we don't. And even in those where we have sophisticated models, they are sophisticated from a scientific point of view, but not necessarily in terms of the level of exposure to regulatory testing that they have received. And therefore, there is still a work that we need to do as scientists in reaching out to both industry, but particularly to regulators, working with industry to raise the trust and the confidence, the credibility on those models. And then the last point is the insufficient expertise in regulators, and also if you go to the next one, also in industry, although lesser in industry than in regulators. So there is a work to be done as well in terms of advocating, training, and working with the whole ecosystem in sort of making these tools to be understood. And these are the premises of the work that we are actually doing in the network. We also asked them if you had, so basically the question was, we gave them 30 virtual coins and we said how you would invest your money if that was at your side into these 5 areas. And first And last, for some reasons, came the importance of standardizations and best practices at the international level and at the UK level. So there is a preference, fortunately, for harmonizing in standards as well. But then if you look at the internal ones, is the need to have a network of UK national capabilities, which is what led to the creation of the CERSI, but also a gateway for access to data and models that are validated, and also to modeling and simulation tools. So that might be also an area where we can work with BioRim, is can we develop some sort of reference data sets in the respiratory space? And then finally, the last question was, which are the stakeholders that we need to reach first if we want to increase adoption? And by almost unanimous support was the importance of working towards regulators and policymakers, which is something we've been doing. And there has been all kind of approaches to do this, including in 2 weeks, I'm actually talking in a parliamentary breakfast, emphasizing as well this in Parliament in London. So to take things forwards, you need to co-design this form with the whole ecosystem. And, basically, a lot of the actions, I'm not going to go through in details from those, but they are fundamentally, arising from those identifications by talking to the community, and also the design of the center as a public-private partnership is in this spirit. And what I would really, really love is that BioRim becomes, one of our vertical sort of deployment areas where we can apply all of these technologies in the respiratory space, and hopefully that's what we plan to do. So I will leave for my second part a bit to talk more about the pilot phase. So this is the next 12 months of the network, but it's the first step towards this vision, right? So when I explain what we're going to do in the next 12 months, it will sound that it's only a part of it, but it will be the first step towards that vision that I described. And my invitation today is whether we can identify some possible case studies that we will incorporate in our pilot, pretty much the same way that we're going to do it with some of the others that I will explain to you. Thank you.
Thank you, Alex. That's really interesting and really set the scene for what's going to come next. So I'll invite Salman Siddiqui, Professor Salman Siddiqui, to share his screen.
I'm just going to share my screen. Wonderful. Thanks, Alejandro, and really delighted actually to be talking just after you. And I thought long and hard about what to talk about for this session. But what I thought I'd talk about were the challenges that I think are unique to in silico modeling in respiratory medicine, actually, because all disciplines are somewhat different. Whilst there are commonalities, there are also particular challenges in respiratory. And I thought that many people that have probably dialed into this webinar do not know what types of in silico modeling have been applied to respiratory medicine and what we can learn from them. So I'm going to give you a whole series of case studies of publications and study results that have used different types of in silico modeling in the respiratory space and highlight what we've learned from them. And I think that should provide a very good discussion basis for discussing how we can really go towards that much higher ground that Alejandro pointed out, which was really around taking these technologies towards implementation, adoption, and into the post-regulatory space. So I'm going to briefly remind you all, and I know you don't need a reminder, about the burden of respiratory disease. I'm going to tell you in one slide why I think respiratory disease poses unique challenges to in silico modelling. It's not cardiology, and it's not bones and joints, but it may well be similar, and I think that is a very exciting space that we should explore as this narrative evolves. And I'm going to give you some really interesting and simple published examples of different types of in silico models in respiratory disease and really share some clinical insights of what I've learned from them. So for those of you that are not clinical, respiratory disease is the biggest non-talked about ticket item in global health. And that's because it affects 1 in 5 people. It's the 3rd biggest cause of death in the UK. It's highly, highly underfunded compared to other specialisms. This is a known historical challenge with respiratory disease. And the public perception of respiratory disease is one of poor awareness, basically. So we've got a lot of work to do. These statistics play out at a global scale. Respiratory disease causes as much disability as the big hitters, dementia, type 2 diabetes. And one of the biggest frustrations of this disease is that it's often diagnosed very late in the disease trajectory, whether that's lung cancer. We have some of the worst outcomes in Europe for respiratory health and lung cancer, late diagnosis, whether it's COPD, where we often diagnose late when the lungs are already extensively damaged and there's very little option really to reverse the remodeling of, that's occurred in the airways. We diagnose the disease late. So we need better diagnostics and we need better approaches to understand the early pathogenesis of disease so that we can really move the needle in respiratory health and improve patient outcomes. And a critical point is that the respiratory diseases, generally chronic respiratory diseases, are a consequence of our genome, but also the interaction of our genome with the environment. And this gene-environmental interaction plays out over our life course to drive respiratory disease. And there are several wonderful publications by our leading respiratory charity, Asthma and Lung UK, that really highlight some of the unmet needs in terms of economic unmet need on the left, in the middle, diagnostic unmet need in respiratory health, the impact that respiratory disease has upon the winter crisis. We had a quademic this year. Every year, respiratory admissions cripple our hospitals, have an enormous impact. And from the government on the right-hand side of my slide, looking at statistics related to air pollution exposure and cardiorespiratory health, as well as death, basically. So going in silico in the lungs, what are the challenges? And this is from a clinical perspective. Well, the lungs are a complex system. They're designed to match ventilation with perfusion, and unlike the heart, where you can actually access the coronary arteries and put a stent in the coronary arteries, or your hip joint, where you can access your hip joint and you can put in a dynamic hip screw and fix it when it's fractured, you cannot access 70 to 80% of the surface exchange area of the lungs without highly invasive approaches such as bronchoscopy, lavage, alveoscopy, which are rare techniques that most people do not have access to. And that's critical because most respiratory disease, chronic respiratory disease, is conceived in the lung periphery, in the very small airways of the lung. And it stays there for many years and smolders and progresses before it becomes symptomatic in patients and expresses itself. In silico medicine in respiratory disease has a historic grounding in lung physiology, actually, and omics, such as genomics. And whilst there have been fantastic studies looking at structure and function with lung physiology, with modeling, and fantastic studies linking omics, such as genomics, to traits such as lung function using statistical association, they're like two trains moving in the opposite direction. Well, what we actually need are mechanistic causal models that link what's happening in cells and tissues to the physiome, to the ventilation and perfusion match or mismatch that's occurring in this complex system to understand these diseases. And we do not have that in respiratory disease. And another point that's critical is that, again, I keep mentioning cardiology, as it's our neighboring organ, right? The heart is easy to access. It's full of devices, so stents, implantable loop recorders, defibrillators. You saw the example from Alejandro about left ventricular assist devices and heart failure. But the lungs, we don't really have many devices for lung disease. We have valves in advanced COPD, we have bronchial thermoplasty in asthma. Some inhalers are smart and they have digital acoustic sensors and time stamping of when people took them, but actually for our most common devices, which are actually inhalers, we don't use any model-assisted simulation. And most inhaled particles actually hit the back of your throat when you take an aerosol inhaler. They don't get into your lung. So we could do a lot better. This is a huge opportunity for respiratory medicine. So there are so many models. I had to look up what the definition of in silico medicine was for this talk. There is no clear definition, but it includes all of these different types of models, statistical association models, clinical triage and risk models, the types of models that I mentioned earlier, so models looking at the physiome, such as gas exchange models, structural models of the lungs, the airways and the vessels, and how the structure then affects the function, and systems biology models that focus on interactions. All of these are in silico experiments and in silico models. That's the definition from Wikipedia, actually, which I thought was quite useful. So they're all in silico models. So I think one thing that we do need to do for some of our key clinical stakeholders is actually bring them up to speed with what an in silico model is. I think there's a vastly different perception amongst different stakeholders. So I'm now just going to give you lots of examples of published in silico models in respiratory disease and kind of what I learned from them, actually, and what we learned from them as a community. So I'm going to start with COVID. You know, during the pandemic, I remember being on call right at the beginning of the pandemic. This was before there were any randomized controlled trials in acute COVID. We had no idea how to treat acutely ill patients with COVID, and they were being wheeled in hypoxic with very little evidence of lung damage on their x-rays, but severely hypoxic. We'd never seen it before. We saw people with oxygen saturations in their 70s with nothing on their x-ray, and we could not understand what was happening. Here's a lovely paper from Tyler Winkler's group with such a super simple model on the left-hand side, a 12-compartment model of the lungs, where the black parts of the compartments are the ventilated parts, and the grey parts are the damaged parts. And you can see that there's a capillary bed going into these compartments where deoxygenated blood is coming in, so mixed venous blood is coming in, and then arterial blood, oxygenated arterial blood is coming out of the system. And in a normal lung, when the lung is damaged, in the grey areas, the low oxygen tension drives vasoconstriction, so you get hypoxic vasoconstriction, so the blood is shunted away from the damaged parts of the lung to the perfused parts of the lung. And so we speculated at the beginning of the pandemic that the reason why this was happening was that there was some kind of failure of the normal hypoxic vasoconstriction that was occurring in people, the normal physiology that we had never seen before. This was a plausible way of explaining why people were being wheeled into our emergency departments with oxygen saturations in the 70s, but nothing on their chest x-ray. And they were talking to us shortly before they had a cardiac arrest, of course. And on the right-hand side are just some super simple models, you know, explanatory models. And these explanatory models are looking at low levels of lung damage, so kind of 15% lung damage, so one segment damaged out of these 12 segments in the lung. And here you can see three scenarios: normal hypoxic vasoconstriction in the blue, impaired hypoxic vasoconstriction in the red, and then reversal of hypoxic vasoconstriction in the orange. And you can clearly see that the reversed hypoxic vasoconstriction, where actually the blood is being shunted through the small damaged region of the lung, even though it's only 15% of the lung, you get profoundly low oxygen tensions. They also went on to show that in the normal lung, if you also had on top of this problem perfusion defects or ventilation in homogeneity in the normal non-damaged lungs, you could also get quite significant hypoxia. And we of course saw in the pandemic that heparin, you know, in patients with a hypercoagulable state, was useful in patients on critical care to improve outcomes. So such a simple model gave us such useful insight into the mechanisms of hypoxia in a group of people that we'd never seen before. Was it translated? No. Could it be developed further for a useful in silico simulation tool when people come into an emergency department based on the level of damage on their imaging and their x-ray and their PA-FiO2 ratio and their response to oxygen? Absolutely yes. So brilliant model hasn't been translated. I'm going to keep going on, and the story is always going to be the same in every single slide. This is another piece of work. This was from our own group during the AirCron program, which Bindi was part of. This was a work program led by Himanshu Kaul, who's now got his own lab at Leicester in emergent engineering, looking at using an agent-based model that was describing the immunology of asthma to really understand how cells, immune cells and stromal cells interact in asthma to drive asthma disease biology. And in this particular study, Himanshu looked at using this model to understand the anti-inflammatory effects of a drug that targets CRTH2, which is a prostanoid receptor expressed on type 2 immune cells and is important in their migration to the lung. And he went on to show on the right-hand side, I'm not going to go through all the specific details and nitty-gritty of the biology, that this model was quite a useful way of predicting why certain effects in the clinical trial, in this case, a reduction in airway smooth muscle mass with this drug, were occurring. And in this case, Himanshu showed that there was a significant relationship between the influx of eosinophils into the lung and airway smooth muscle mass, and that the drug might be actually reducing airway smooth muscle mass by targeting the migration of eosinophils to the lung, although that was only one of the mechanisms that could explain this. Very useful model. Has it been translated? No. The code sits in a very specialist software environment. Other groups have yet to use this, but could this be useful in asthma immunobiology studies? Yes. Third example, I'm going to rattle through these examples now because I really don't have a huge amount of time. These are patient-based models of physiology. This is Brody Foy's work, and Brody's since also got a chair now. He's at the University of Washington, and he has a chair in math modeling. He started off in the AirProm program. And this is work taking patient-based images at the top of the screen, extracting their airways using segmentation, growing out the complete conducting airway tree, and simulating acoustic physiology of the small airways to understand small airways disease in the lung. At that time, acoustic physiology wasn't popular at all. Nobody liked it, nobody used it. Now it's suddenly come into huge vogue in respiratory medicine. At that time, we were able to show that narrowing and obstruction of the very small airways was important in driving these acoustic physiological abnormalities that we thought were related to small airways disease. And we then used these models in the context of a clinical trial to infer how a treatment relative to a placebo could impact on small airway obstruction directly when we hadn't measured it in the trial. Useful technique. Has it been translated? No. This is another study using the same technology. This is Min-Suk Kim's work. He was also part of AirProm. He then moved to Oxford to Fergus Gleason's group. How many people have a CT scan of their lung? Lots of people. And how much information is extracted? Hardly any. That's a standard CT scan. That will get a radiological report if you go into your hospital with a breathing problem. That's a xenon imaging scan, a specialized scan to look at the ventilation in the red and the non-ventilated parts of the lung in the blue. It's imaged using dual energy. I'm not going to get into the details of it. And this is just a simple, complete conducting airway tree model called the FAN model, which Min-Suk developed, simulating the distribution of xenon and the ventilation using the geometries and the tissue densities from a standard CT scan. Look at how good the concordance is just using a simulation. You can get really detailed spatial ventilation maps and a near line of unity comparing the xenon ventilation with the ventilation from simulated model in COPD. This could transform medicine. You could identify small airways disease using routine non-contrast CT scans from big programs like lung cancer screening. Has this been translated? No. Medical devices. This is a study that Bindi led, lovely study. And we used to do lots of camera tests, putting tubes into airways, heating the airway through a technique called bronchial thermoplasty. We heated the muscle to 65 degrees C in an airway to zap it and shrink the airway smooth muscle in asthma. This lovely study using COMSOL-based modeling, using a bioheat model, went on to demonstrate that actually when you heat standard airways with the bronchial thermoplasty catheter, you don't really get much heating to 65 degrees in the majority of the airway wall. It's only when you get right to the distal tip of that catheter, the smallest possible airways that the catheter can target, that you get this uniform, even heating of the airway. And actually, a novel hypothesis that could arise from this study is that actually if you redid the thermoplasty trials, giving more activations in the more distal tip of the catheter, that you might get better therapeutic effects. Very important hypothesis because we've stopped doing this technique in the UK largely because we don't know how it works. Final couple of slides, these are biostatistical models. We're taking normative patient populations using Bayesian modeling. The group have extracted patient trajectories of lung function decline. And then they fitted new populations, in this case with COPD, to those decline trajectories using baseline characteristics and lung function. Is this useful? Yes, you could tell a patient who's smoking, who comes to your clinic at the age of 45, that they've got a 90% probability of being on the green line. That could be a very powerful incentive for that patient to actually stop smoking. Do we use these trajectory graphs in the clinic? No. Final 2 slides. Drug discovery. This is my favorite paper of the last 3 years. This is a wonderful paper showing the power of AI in drug discovery, identifying a new small molecule inhibitor that this group got through conception, through to molecule generation, lead candidate identification, all the way through to phase 1 in 18 months. Absolutely astonishing. Potential new drug now in phase 2 trials for idiopathic pulmonary fibrosis, a major scar in lung disease. I really encourage you to look at this paper. I think this is extremely exciting. Multi-scale models, very complex, sophisticated models linking the organ scale effects in tissue to the tissue scale to the organ level, helping us understand actual disease mechanisms in the tissue play out at the organ level. There have been very few publications using these truly multi-scale models, but I think that they could be transformative in helping us understand complex chronic respiratory diseases. So to summarize, we're clearly at the advent, as you've seen from these publications, of the kind of intersection of these clinical prediction models using statistical modeling, but also these much more complex physics-based models that cross the scales or exist at individual scales. And I think we'll see a convergence of these 2 technological approaches over the coming years. And yes, we can get regulatory approval in respiratory disease. Here's an example with an imaging tool to look at lung ventilation response using CT. This is called FRI, or functional respiratory imaging. This has now been approved by the FDA. The company is called Fluida. So I'm going to summarize. Going in silico in respiratory, there have been amazing studies to understand lung physiology, structure, function, and now drug discovery, but very few have actually gone through regulatory approval or have progressed that translational bottleneck. I think it's super exciting that there's an emerging class of multi-scale and multimodal applications that really, I think, will transform our area. But the big but for me as a clinician is that nobody's going to use them and nobody's going to trust them unless there are standards like Tripod AI for how you report these models, how the code is deposited, for how you could train people to use these models. And ultimately, clinicians need to be able to use these models in the clinic. Otherwise, they will never get disseminated into practice. So they have to be usable by a wide group of stakeholders. And I think those are the key challenges, really, and perspectives for respiratory. Thank you.
Thanks very much, Salman. That's really eye-opening, especially for lots of us who don't see the clinical perspective, perhaps. So I will move on quickly and ask, I'll share my screen, but I'll ask Anne to perhaps, the project manager for Barium, to just talk to us a little bit about the next steps. Shares my screen, sorry, I'm being a bit slow here. Can we see that okay? Can you see that okay, Anne?
Yeah, thanks, Bindi.
So yes, so following on from this webinar, what should you expect? And so Anne, if you just talk us through the timeline, I will then hand over to Alex after that.
Thanks, Bindi. So following this webinar, this is really the first in a 3-part series of our ongoing work with In Silico UK and CERSI. So we hope that this is, the start of building up a kind of evidence base, and we hope to build up use case studies specific to respiratory, of examples where I've, in silico technologies exist, or whether there's an unmet need that could be addressed by in silico technologies specific to respiratory. And so we are putting a call out to our community to submit ideas for example use case studies following this webinar, which, as you can tell by how much content we've had in this 1 hour webinar, there's a lot more to interrogate into that, and a lot more discussion around these example areas. So we are hosting a 1-day workshop on the 14th of March in Manchester, which we invite you to join us on, and we'll be sending through the details after this webinar of how to register and where to submit those ideas for use case studies. And that's where we'll present some ideas of these use case studies, interrogate into them a bit more, and look at how we can build up that evidence base specific for respiratory. And there is some funding through the BioReam network to support those onward activities, building up those case studies. And we will finish the event series as it is, with a final event in Manchester again on the 9th of September as a stakeholder showcase event to showcase these use case studies and to strategize our next steps forward. I think maybe in the interest of time, Bindi, it might be best to go to the Q&A.
Yeah. Okay. So I think we will perhaps just hand over to Alex, who can talk us talk us through the details of what we mean by use case studies, but perhaps through some exemplars of his own, I believe. So Alex, let's do you want to share your own screen?
Yes. So I think, are you able to see it now? Okay, super. So,
okay, thanks.
Perfect. So thanks a lot, Bindi, and it was really great, Salman. I think that list you produce is a great starting point for identifying use cases, and I think will become very handy to what I'm going to explain. So when we met for the first time, Bindi, Salman, Anne, and I, it was a really fortunate situation where we were at the timing that was very, well, it looked like hand in glove, right? So what I'm going to explain you now is a process that we're going to be running in the CERSI, where unfortunately there is no respiratory use case. But fortunately, this is an opportunity for BioReam to actually help us to ensure that this is well represented. But what I will explain you is what the whole idea of the pilots or the case studies is. So as you remember from my first talk, I explained that the key thing that came is the importance of credibility assessment. So going from where we are to where we need to be requires that regulators trust the results of models. And this is going to be the focus of the next 12 months, is what we call an in silico regulatory airlock initiative. We call it an airlock initiative because it's a safe space with regulators, clinicians, academics, and industry can work together to develop a better understanding about what are the barriers, the practical barriers in very concrete examples, and develop best practices that are both specific for those case studies, but by looking to multiple of them, they have some sort of principles can be distilled from that. So what the focus or the kind of criteria for a case study is, it needs to be of pre-competitive nature so that everybody can have a conversation, pre-commercial. So we are not going to be talking about specific products. So we may talk about a CPAP system, but we're not going to be talking about a particular product that happens to be a CPAP device. There has to be some model that we want to look at, and we want to look at the credibility. So I think Salman was saying that those inhalers have a problem in the way that some of the drugs get in the airways. Well, it may be that the problem we want to look is at the proportion of inhaled drug that actually gets to the airways, and there is some sort of model that is a combination of computational fluid mechanics with some sort of dispersion. So you need to identify what the model is and what is the interesting question of regulatory value. And we're going to use those And what we're also going to do is we are not going to implement the use cases, in the sense we are not going to implement the models and run them. It's going to be a thought experiment, because otherwise this can be a 3-year project, each of them, right? But what we are going to do is try to understand what are the regulatory barriers and what are the implementational barriers if we were to actually go and do it, right? So in a sense, it's a feasibility or a thought experiment that will hopefully lead to a number of projects that could be submitted for further funding. And but they all need to have some sort of regulatory impact, and that's why you will see that we want regulators involved. And they will also give us the opportunity to look into sort of social technical considerations. We believe that this is not just a technical transformation, it's also about what patients think about this evidence. Imagine yourself as a patient and you have the next device has a label that says this has been validated with numerical methods, with no patient, right? Would you take that medicine? Would you take that device? So what are the sort of public and patient views of things? So we worked together with MHRA, and they provided us a set of guidance, which again, I'm not going to go in detail, but the whole design is based on answering to those, and you will have access to the slides. And we also have made a similar work to the one that Salman did in summarizing use cases in respiratory. I'm summarizing here some of the key guidance that are internationally around this area. So there is already some FDA guidance, albeit they are relatively new. And there is also some standards by ASME, by the American Society of Mechanical Engineering. There is a number of standards with ISO that are being developed as we speak, okay? So these use cases also will feed into that standardization effort and will nurture from those. Now, the gist of some of these standards is that there is a reality of interest, the patient and the device, or the patient and the pharma, the drug. There is a mathematical model, which is kind of the abstraction of the principles into a set of equations. And then there is a computer model, which is the implementation of that physics or that biology that we are interested in. And we need to understand how all of the things interrelate to each other. And this is what essentially the ASDME standard helps to do. So I'm putting here an example of you have a virtual model of the heart and a virtual device and the real entity. There is a real trial and our computational trial, and you need to go through a number of questions that this standard helps to decide. But fundamentally, all start by what is the question of interest? I want to know the amount of inhaled drug that actually gets to the airways. What is the context of use? I want to use this to answer a particular paragraph in my regulatory dossier. And then what is the risk? How much I rely on the model versus also relying on other information of experimental nature. If the whole regulatory dossier was to be based on model, this is a high model influence. And what is the worst thing that can happen if my model is wrong? This is the decision consequence, okay? So I'm just summarizing. There's a lot of documents about how to do this. So I'm not intending to leave you all the details, but just that you know that there is a lot of work already done. So what we are going to be working through those case studies is in helping to develop for MHRA a position paper that we think is going to be the seed of a possible guidance. Okay. Now, in our project, what we have is a number of committees. There's a number of committees, 4 committees, and one of them is a technical committee that I would like someone from the BRIM leadership to be part of, so that you can provide as well oversight to the other pilots that are going on. And there is a number of 10 case studies that we're going to be looking at. And these 10 case studies, there is, forgetting the first one, which is in manufacturing, there are 3 in cardiovascular, 1 in neurotechnology, 3 in musculoskeletal, 1 in related to drugs and in the context of COVID. And then there is 1 in radiotherapy. And hopefully there will be an 11th on the respiratory or the 12th in the respiratory space. With each of those, we're gonna give the opportunities of a 2-hour session online, where the team that will be built around, say, the pilot 11s on respiratory will have to have at least 7 members, 2 that come from industry, from 2 different companies, 2 that come from academia, from 2 different academic institutions, 2 that come from regulators that we will help you to identify, and 1 ideally that comes from the public, that will be kind of your public rep. So each of these 2 hours will the first half an hour will be a presentation of the case study to the rest of the community. So like Salman did, but instead of presenting 5 or 6 of them, you will take one and you will explain it in more depth, why you chose it, what is it, the impact it has, etc., etc. You will then spend another half an hour explaining what is the process you follow and the lessons you learn in that case study. And then we will have up to an hour where will be a Q&A by the rest of the community. That's you will have people from cardiovascular or from musculoskeletal that will ask questions to you to try to help to understand the relationships between different case studies, or will also challenge you in some of the approaches that you took so that you learn as well from others. And you will have the opportunity to sit in all of the other case studies, so you will be able to do the same with the rest of people and learn from them as well. So my proposal, and the reason why we have this final meeting in September, is because this project will run, sorry, this is, yeah, that's correct. So this project will run actually a bit longer. We have until January 2026. So my proposal is that after September, when we have this meeting, we will look for a date where we can run that case study that comes from the respiratory space. So you have the opportunity to see all the other 10, in addition to have time to prepare them properly. That will feed into a number of other things we are doing, which I'll save you now. And then finally, from a very practical point of view, there is already some documents that are written where what exactly a pilot is, the composition of the teams, what is expected is already described. There is also a template for how for you to write essentially a first case study. And then there is an example that we give as a prototype for you to work from. And all of this is in our Silico UK publication repository. And this is basically the description. It has been already downloaded quite a bit. And this is the link that I will put on the chat in a moment where you can take it from. And we have also some descriptions of different roles and responsibilities for each of the members, if you are interested in, that can help to get this organized. So you get all this framework that you don't need to kind of develop from scratch. So I think that's it. I would say I put on the chat also the link because there is a lot to learn and none of us has all the expertise, right? So we are going to be running a number of webinars in the next few weeks that might be of interest to you. And if you join our community, you will get access to all of that and to all the announcements. And these are some of the courses, but again, this will be announced and informed through our channel. So I will leave it there. But I understand that from the conversations we had, the idea would be that between this and the next meeting that we will have in person, everybody who is interested in this grouping to put forward case studies, that you reach out to Anne, to Bindi, to Salman, to myself, and we'll then sort of we'll Sherpa you through the process to facilitate things. Thank you.
Thanks, Alex. Can people see my screen? Can you hear me okay? Can you see my screen?
All good.
Yep.
Yeah, okay, great. Sorry. So yeah, so before, just a quick thing really, we had planned for a question and answer session. If you're able to stay online and ask those questions, then please do. But just a quick note here to say that before we get to that very well developed use case study that Alex described, we do have this series of events that we're organizing as BioReam to develop that, to sort of input and develop those use case studies. So the first step will be to complete a soft touch survey, which sort of allows you to identify or suggest some use case studies first, which we will review and then invite you to come and present at a sandpit, at which there will be lots of input and advice and help on how on all of the things that will be needed for this airlock to be included as number 11, say, within Alex's framework that he described. So I will just perhaps make sure that I've covered things that I need to say. Yes, these are some of the things that Alex has already mentioned. And then I will perhaps finish there and invite people to ask questions and ask Terry to moderate. the session. There might be some questions already that you can.
Thank you, Bindi. I'm thinking that we are running short of time, and I've seen that Salman has to run out for another call. Well, I'm not that much surprised with two very passionate speakers like today. I need myself to jump to another element, but nevertheless, I've got a question that was coming. One of them that I like to ask to Alex right now. Alex, when you say simulation first, do you mean that it should become mandatory to do simulation before doing any kind of animal testing or clinical trial in the future?
I think the short answer is yes, unless you can demonstrate that it's not possible to do it. It should be, my view is that you should demonstrate that something is not possible, it doesn't make sense, there is not the basic science or the models available, because there will be situations that won't be possible to model. But if you could, I think there is an ethical imperative of why you would carry on and do all the other things before you even check whether this is feasible or not. And at the moment, I think the reason why that doesn't happen is because we don't have the tools, you know, and the credibility, the access to data, and the expertise that is required to do this at pace. But I hope that this is something that we will be able to change as a community over the next few years, right?
I would fully concur to that. We need to make it mandatory. Otherwise, people might still be thinking, okay, they can do it otherwise. I'm convinced that as soon as they will start doing this kind of in silico testing, they will see the power of it, the power to learn quite a lot of information, and therefore they will get the taste of success of it. And we'll see that this will continuously increase. But unfortunately, like always, it's always very important to impose some things for the sake of the industry and for the British citizen. One question that was asked to me before this meeting, and maybe because in my position as President and Secretary General of the Aviation Alliance and City of Healthcare at ANSYS, I'm also an e-health expert to the European Commission. The question was related to compare the different kind of landscape. What is the difference between the landscape in Europe and in UK? I would say that from my perspective, the situation is much more agile in UK compared to Europe, because UK is a smaller region, but it's also a simpler situation. And of course, you've got a lot of support from the government, which is still quite a little bit challenging right now. We know that many of the large companies are anxiously looking at Europe because this is a very large market, but nevertheless, using UK as a proof of concept could be very valuable, not just to encourage other countries to follow that, but first of all, for the British citizen, will benefit from this treatment much faster than any other citizen in the world. And of course, for this kind of British industry that could accelerate this kind of medical innovation. So that's a big difference. You are going much faster than in Europe, but with my corporate perspective, I'm glad to see that, because this is also a way to challenge Europe to say, okay, what are you doing over there? And they're taking it quite positively. One last question that I would ask to Alex here. Can you confirm, because it's sometimes not always very clear, can you confirm that today we can submit in silico evidence for regulatory approval to MHRA?
Yes, the short answer is yes, you can. The current regulation in the UK doesn't mention explicitly, but also doesn't exclude any form of evidence, in fact, including, you know, in silico. The only thing you need to demonstrate is the only thing that it does is that the evidence needs to show that your product is safe and efficacious, and also, of course, you need to show that your evidence, whether it's in silico or otherwise, is scientific, so that it is sound, that is rigorous. So that's the standard at the moment, and this is something we've been discussing with colleagues as well in the regulatory agencies. And in fact, they're very open for you to reach out to them and discuss a particular study and basically take their advice from early on.
And is the ASME VVUQ 40, although being an American standard, the one that is recommended to be used in UK to validate and verify the model?
I don't think there is an exclusion of the standard, but there is also not an explicit adoption of it. And that's why there is a work going on to raise it to the levels of, to an international standard, working with ISO. And I'm actually part of the working group that is working in that effect. So if anybody is interested in engaging, particularly from industry in the standard development, if you drop me a line, I can tell you whom to reach out to connect. I don't think there is representations for every country, but there is representation from quite a number of them by now, from both Europe and the UK. And BSI, the British Standards Institute, is representing the UK there. So there is, when that standard happens, it will be a standard for the UK as well.
Thank you very much, Alex, for this answer. And then, Bindi, I would suggest to get back to you right now because we are already a few minutes late for all the audience, and I certainly want to thank the audience for staying so late with us.
Thanks very much, Terry, and thanks, Alex, for both of your contributions to today. So yes, I think we should end that there because we are 10 minutes over. If you have any further questions, please reach out to us. There are the other events that we've got planned, and we are happy to provide further guidance on all of the things we've said today. And this presentation, this webinar will be made available online as well. So with that, I thank you all. Thanks very much for attending, and we hope to see some of you in the events to come.